right, we're into chapter 4. That is part of unit 2. So 1 through 3 was unit 1. Chapter 4 is in unit 2. And we are going to start talking about cells. So we've talked about macromolecules previously. Now we're going to talk about what happens when they come together to form our basic unit of life, which is the cell. So we're going to talk first about how you study cells. There's some problems to this. They're kind of tiny. That causes issues. We'll then talk about prokaryotic cells. We will do a little bit on eukaryotic cells after that, actually quite a bit, because we're eukaryotes, we care a lot about them. And then we will talk about inside of eukaryotic cells, this thing called the endomembrane system and some of the proteins involved there. We'll talk then about the cytoskeleton. This is kind of like a rail network and structure inside of a eukaryotic cell. And then we'll talk about connections between cells in multicellular organisms. So cells, they're our basic unit of life, but we often in multicellular organisms see very specialized cells. You have eyeball cells, liver cells, brain cells, skin cells, cells that do special things. So we're going to talk broadly about cells, but not all cells are going to fall into the things that we'll talk about. Like, for example, let's look at some different types of cells. The cell is the basic unit of life. Here we have human tissue. This is probably skin here. We probably have epidermis here. And then some of the cell types, keratinocytes and things like that, that are below the epidermis. Here's a plant epidermis. We have stomata here with the guard cells there. Did some undergraduate research on these. I have a fondness for them. These are called pavement cells. They kind of lock in the water inside of the plant. And under them, there's other types of cells specialized for photosynthesis. Here are some fungal cells with some hyphae. And then these are probably spore-forming cells here. Here's red blood cells. They're part of our body. And in this slide, we have some trypanosomes. These are parasites that are in there. Now, I said not all cells are going to be identical here. Red blood cells are kind of funky. They're really specialized for carrying oxygen. Red blood cells actually lack a nucleus. Even though they're part of us and we're eukaryotes, they have a very specialized structure. So some cells are super specialized. Neurons and things like that, they're real specialized too. We're going to kind of keep things basic here, and as you go on in your specializations, you'll learn all about fun special cell types. Of course, microbes, we have different bacterial cells here. There's a whole mix here. We have some gram-positive rods here that stain purple, gram-negative ones that stain pink, and these dark purple spherical cells. Those are cocci. Those are diplococci there, and they may cause like meningitis or something like that there. So the cell, though, whatever the cell is part of, that's our basic unit of life. That's why we said that viruses weren't living. Remember, within eukaryotic cells, we have organelles. So like the nucleus, or this thing called the endoplasmic reticulum, mitochondria, things like that. These cells might arrange themselves into tissues. We're not going to go to that level here. We're going to focus just on the cell itself in a general sense. In prokaryotes, these are not to scale, by the way. This would be about 10 times larger than that in most cases. Prokaryotes, on the other hand, they have variable cells with different types of cell walls. But inside of them, they have usually a what we call nucleoid region. So there's no membrane in there, but the genetic material is not just floating around randomly. It is organized, but we also might have some outer layers and some flagellae or pillae that are used for motility or sensing the environment, things like that. So we are going to talk about first how we study cells before we talk about the details of different cell types. All right, cells are microscopic. We need to talk about microscopes here. So we're going to talk about the roles of cells in organisms. We're going to talk about two types of microscopy, light microscopy and electron microscopy. Then we'll talk a little bit about cell theory. Again, I'm going to keep hitting this. If you want to learn more about cellular theory and how we came to kind of determine that the cell is the unit of life, my Micro 250 class, I talk a lot about that in there. All right, so we have a problem, right? How do you study small stuff? Particularly problematic when the small stuff is too small to see with the human eye. That becomes a big problem, right? So you're probably aware, right? Sherlock Holmes, depicted in media, always carrying around a magnifying glass, right? To examine small details and clues there, right? Of course, he also smokes a pipe because it was the 1800s and everybody smoked, right? But how do we study small stuff? How do scientists study small stuff? Real small stuff. Well, when we get to the micro level, like micrometers, we're talking real small, right? Micro means small. We use a microscope. Scope means to look at. A microscope is some instrument that magnifies an image. You are probably used to enlarging images, but if you've ever used your cell phone to zoom in on something, you might notice that at some point the resolution starts going down, right? Your camera on your phone has a set resolution. If you zoom in, yes, you are magnifying it, but you're not necessarily making it clearer. So microscopes have two aspects to them. They have magnification, which means making an image larger, but they also need to increase resolution. Resolution is the ability to tell two close objects apart. So here is a high resolution image, right? We can see all of the curves of these different flower petals, and this one is the sepal, and we can see that compared with the background. Lower resolution image, I can't really tell how many petals are in here, right? It's just kind of a purple blob, so I can't distinguish, well, let's say this one from that one, right? Like, I can't really distinguish those things because the resolution is too low. Resolution is going to be tied to the properties of physics. So if you are interested in microscopes, you're going to need to learn some physics, particularly the physics of optics, which is dealing with lenses and things like that and how light moves. But also you're going to need to know about electrons. Interestingly, right, electrons behave slightly like photons. We're not going to get into quantum physics here, but light is actually made up of particles called photons. They behave both like particles and waves. It's weird. Electrons are particles. They also are wave-like, but particles. That's quantum physics, okay? What we need to know here is that there are two different wavelengths here. So we'll come back to that in a moment, but this is going to limit how much we can magnify things. Light microscopes are pretty good at magnifying things. You can go pretty good, but electron microscopes are going to be able to magnify and resolve images at a much higher level. All right, so let's talk light microscopy first. This is the most approachable. We'll be able to do light microscopy because we have light microscopes. We do not have electron microscopes at CEI. They're very expensive and hard to maintain. So here we have an image that has been magnified, and we have some red blood cells in here. You probably have seen blood, right? In it, you generally do not see red blood cells with your eye. You need lenses and magnification to see that. Just as a side note, there's some white blood cells in here as well, just for interest. So light microscopes are relying on photons of light that come in and either bounce off a sample or pass through the sample. That'll be important here in a moment when we talk about the difference between that. Our microscopes, our light microscopes, their max magnification is going to be about 1,000 times magnification. That's pretty good, but it's actually not good enough to see certain structures that you might be interested in. The resolution here is going to be about 200 nanometers. That's about 0.2 micrometers. That's pretty good, but again, we will find that there are some things that are smaller than that. Particularly in biology, we're concerned with viruses. Viruses are too small to see with a light microscope. That actually makes discovering new viruses very, very difficult. That is one of the reasons that COVID was not discovered right away, because you have to look at it under an electron microscope. The resolution here is limited by the wavelength of light. So light is a wave, and the shorter the wavelength, the smaller the resolution is. So the longer the wavelength, the higher the resolution is. Light is limited to about 0.2 nanometers in terms of resolution there. We also often need to add stains to samples. So here the red blood cells show up kind of red because of the molecules in them, but there have been some stains added here. This dark purple stain has been added to show these structures. Many things that are microscopic don't actually reflect color because microscopic things don't have eyes, so they have not evolved pigments to display themselves. The light microscope we're going to use primarily in our lab is a compound light microscope. It's made up of 2 lenses. We will also talk in lab about what we call dissecting microscopes. They are slightly different. They're for viewing larger structures. When you read the lab, make sure you pay attention to the discussion about dissecting microscopes there. In a compound light microscope, there's 2 lenses. There's the ocular lens, those are the eye lenses, and then these different rotating lenses here that have different magnifications. And this works by shining light through a sample up into these lenses into our eyes. So our sample has to be thin enough that light can go through it. If you stuck your finger under there, no light's going to go through. You would just see a big black blob, right? So we need really, really thin sections, and we're going to shine the light through it. It's going to come through. Some of it's going to get deflected. Some of it's going to make its way into our eyes through these lenses. We'll talk more about microscopy in the lab. Electron microscopy, on the other hand, uses beams of electrons. Instead of shooting light through or at a substance, we're shooting electrons. Now, the benefit of that is electrons have smaller wavelengths than photons, so we get a higher resolution. The max magnification is between 1 and 50 million times, okay? That's much better than our 1,000 times magnification from a light microscope. And our max resolution is 0.1 nanometers. Remember, it was 200 nanometers with light microscopy. So we get way better resolution. There are also 2 different types of electron microscopy. They're kind of similar to our light microscopy. We have scanning electron microscopy that bounces electrons off the surface of something. That's kind of like our dissecting microscopes for light microscopy. Then we have transmission electron microscopy that actually shines electrons through a substance, like our compound microscope we just saw. Unfortunately, there are some drawbacks to electron microscopy. To do this is very expensive and difficult, but also it needs to occur in a vacuum, and generally you need to apply special chemicals to your sample. That means we do not get living samples in electron microscopy. They are all dead. That might not sound terrible. You can see a lot, but sometimes you want to see how things move and change in real time. Video and time lapse has actually revolutionized microscopy. The ability to do this over time and see how things grow and move is very powerful. Electron microscopy can magnify things so much, though, that we can actually start to see molecules. That's where it gets really cool. Here is an image with right here, this little line here, that is an mRNA. And these little balls here, these are ribosomes that are being translating this, and then these are amino acids that are coming off of that. So here we are making a protein off this mRNA. That's pretty incredible magnification, right? So electron microscopy is super cool. And you can get like super high resolution images here, but it's very difficult to do. The theory that all life is based on cells took actually quite a long time to become kind of generally accepted. The problem is that we didn't have lenses for a long time. We had no way to magnify things. So for many, many years, about, most of human history, basically, we had no way of observing the microscopic world. Again, if you want to learn more about this, my Micro 250 class, we talk all about this because the micro is all about microscopes, right? We didn't know bacteria existed until we had microscopes. The first person to see and name cells was this guy named Robert Hooke. If you take physics, Hooke's law for springs is named after him as well. He was very good at grinding lenses, and he built some of the first compound microscopes. He used a candle that shined light through something, and then it went up through his lenses into his microscope. And here's one of his drawings. This is Hooke's drawing of cork cells. He named them cells because there were these little boxes here in the sample of cork, and he thought they looked like the little rooms that monks lived in a monastery, and those were called cells. And the name stuck. Hooke produced a very famous book called Micrographia. There's all kinds of cool digital scans of this. He has beautiful drawings of all these small things. Here's a flea that he looked at under a microscope. He was discovering a whole new world when he started looking at things. Hooke was good, but this other guy, von Leeuwenhoek, around the same time, no relation, he was even better at making lenses, and he could grind lenses so fine that he could use a single lens, and he was able to observe microbes from this, microscopic organisms. People had predicted that there might be organisms that were microscopic for a long time, but it wasn't until von Leeuwenhoek in the 1600s, late 1600s, that we were able to actually see them. All right, here's a bit of a weird story. Von Leeuwenhoek was a dude who worked by himself at night. So he naturally observed the seed of life, let's call it. He observed sperm cells. I'm not going to go into any more detail about how he obtained them, but he did observe those for the first time. That was kind of revolutionary. Like, how does life get transmitted from in sexual reproduction? So he was able to observe sperm cells there. Moving on. In the 1830s, so almost 200 years later, we finally have this kind of unified cell theory that gets proposed by two people, Schleiden and Schwann. One of them was a plant biologist, one of them was an animal biologist. They went kind of like, hey, I see cells in my plants, and the other guy's like, I see cells in my animals. Maybe all life is made up of cells, and that is our unified cell theory. All living things are composed of one or more cells. The cell is the basic unit of life, and new cells can only arise from existing cells. There was a whole other set of experiments that led to that, basically saying spontaneous generation does not happen. Cells don't just come out of nowhere, right? We get cells from other cells. As we continued to progress, we realized that there were two broad types of cells there. Prokaryotes are bacteria and archaea. Single-celled organisms, no nucleus, no organelles. The eukaryotes, on the other hand, many of them are single-celled, but we also have multicellular organisms. inside of their cell, they have a nucleus, which contains the genetic material, and they have other membrane-bound organelles in there. We will talk in detail about the components inside of these cells in the coming sections. Okay, so the cell, that's our smallest unit of life. We briefly talked about how we know that and how we discovered them. You got to use microscopes. We've grouped them into prokaryotes and eukaryotes, depending on if they have a nucleus or not. To study cells, you need a microscope. Magnification is important, but also we need resolution to see an image clearly. Light microscopes can magnify pretty well, but only up to the resolution wavelength of light, about 1000x and 200 nanometers. Electron microscopy, much higher, millions of times magnification, and like 0.1 nanometers. So very, very good resolution. If you want to look at viruses, you got to go to electron microscopy to do that. You cannot see them with light microscopes. These observations led to a unified cell theory that all living things are composed of cells. Cells are the basic unit of life, and new cells can only arise from existing cells. All right, that's it for 4.1. In 4.2, we'll talk about prokaryotic cells.